Welcome, everybody, to Harvard University's Museum of Natural History. My name is Peter Gerges. I'm on the faculty here in the Department of Organismic and Evolutionary Biology. I am a deep sea biologist. I study microbes that live in the deep sea as well as animals and the role that they play in shaping our natural world. A few years back, I had the fortune of working with Professor Pam Silver's lab on thinking about how we might harness the capacity of microbes to eat electricity and produce a biofuel. That is when I first met Christina Agapakis. So I wanted to tell you a little bit about her today. It's my great pleasure to be introducing her. She did her undergraduate work at Yale University in molecular, cellular, and developmental biology, after which she came here to Harvard. And she worked on her PhD in biological and biomedical sciences in Pam Silver's lab. As I mentioned, that's when I first met her. And one of my earliest memories of you, should I share this? You don't know what I'm going to talk about, do you? No, it's harmless. But we had a lab meeting, and we were talking about whether or not it would be possible to make this one organism that did none of the things that we were asking it to do. It just didn't do them. And the question was, could we get it to work? And I was really impressed by Christina because she stood up and spoke up and said, I think we could get these 3 out of 5 things to work, but these other 2 things, I just don't really know. It may be too much to ask of this particular organism. And it was neat to hear a graduate student very respectfully, but confidently state, we should really give this some thought. And I think that was a really neat thing to see. Certainly in a world where sometimes we bite our tongues because we don't want to, we're afraid of looking foolish, it was great to see Christina not be worried about looking foolish. And I mean that as a compliment. I really do, honestly. No, but in all seriousness, though, her thesis was entitled Biological Design Principles for Synthetic Biology, and it was fantastic in that it centered on identifying and utilizing design principles for bioengineering, thinking about the evolutionary and ecological context of organisms before, again, you just sort of dove into trying to get it to do something for humankind. She worked to engineer photosynthetic bacteria, to invade animal cells, kind of giving animal cells chloroplasts, which was so cool, and really reshaped the way we think about how a cell interacts with things like chloroplasts, or what we might call organelles. So it was really exciting stuff. And her thesis really covered a range of work she pursued during her doctoral career, right? She also mentored the Harvard iGEM, or International Genetically Engineering Machines Competition. It's a team of undergraduates that Christina led in 2010 to develop an open source toolkit for plant engineering. It was known as Harvard's iGarden. She later worked on one of my favorite projects, a project called Self-Made, and she focused on the microbial ecology of cheese and other things. Moving on, she then went to UCLA, my alma mater, and did her postdoc there. While she was there, she was also a fellow in the Department of Design and Media Arts. She's now the creative director at what I would argue is one of the most creative biotech companies around, and that is Ginkgo Bioworks. It's known as the organism company. And what they do is they look to nature for inspiration on what organisms are doing and which organisms are best suited to do the kinds of things that we want them to do, right? To work in collaboration with them as chemical engineers, to engineer organisms, to have new capacities, that allows humans to do things that we might not otherwise be able to as easily or do in an eco-friendly manner. But while she's there, she's not only specializing in the sort of organisms like yeast and bacteria, but doing a myriad of other things. She's also the recipient of many accolades and awards, including being a Forbes 30 Under 30 recognition, I suppose, the L'Oreal USA Fellowship for Women in Science in 2012, being on Wired Magazine's Next List 2017 of 20 people who are creating the future. She's a fantastically prolific writer and artist, and I don't want to steal your thunder here, but I do want to share with you a poem she wrote for this evening's event. Roses are red and violets are blue. I'm giving a talk tonight about loving the bacteria in your poo. Without any further ado, let's welcome Christina.
Thanks, Pete. That was amazing. I'm like emotional up here. That was great. I do remember that project. Did it work? Did those two things that we thought were that I said weren't gonna work? So yeah, thank you. Thanks so much. Thanks for coming out here on Valentine's Day. I hope that I can bring you some love of bacteria. So I want to talk about E. coli. I love E. coli. They are very, very, very small, but they are mighty because they double every 20 minutes. And they grow so fast and so prolifically that if you were to give them all the space that they would take and all the food that they would need, they would go from 1 to 2 to 4 to 8 to 16, et cetera. Eventually, after about like 23 hours, you could fill about an Olympic-sized swimming pool. 20 hours later, you'd have a mass of E. coli the size of the Earth. And then round it out to about 48 hours, it's 22 times the size of Jupiter. So lucky for us, there's no celestial bodies made of E. coli because these bacteria, though they grow exponentially when they can, they never actually can grow beyond the confines of the space that they're allotted, the context in which they grow, their ecosystems, and the things and the forces pushing against them. So I like this quote from Darcy Wentworth Thompson, who wrote on growth and form in the olden days about this question of scale. How do organisms scale? And he says that the effect of scale depends not on a thing in itself, but in relation to its whole environment or milieu. It is in conformity with the thing's place in nature, its field of action and reaction in the universe. So this is a theme that I'm going to come back to throughout this talk, thinking about what is the place in nature for technology, for bacteria, and for us, and how do we all interact in this complex milieu. When we think about nature and technology, often those are kind of positioned as opposites, right? We have nature that's wild out there, untouched, separate from humans and the activities that we can do, separate from our technology. But as a genetic engineer, I'm interested in this question of what if our technology looked more like nature? What if we were able to work with biology and work with nature to build new and better technology? So you look at E. coli, you zoom in, you see the kinds of things. The reason why I get so excited about bacteria and bacteria as technology is to look at the intricate complexity of all of the stuff going on inside of the cell. You have this insane, atomically precise machine that just grows itself in poop. It's amazing and so remarkable when you compare it to what it is that we can do as humans as well. So when you zoom in even further into this is the flagellar motor, so like those sort of hairy things off of the E. coli that E. coli used to swim around. That's the motor that's attached to the membrane of the cell, and that really is a motor. It's like a 30,000 RPM motor that spins around. It's made out of a bunch of different proteins that sort of self-assemble into this large machinery. And overall, it's about 40 nanometers across when you look at it under the microscope. When you compare it to human technology, this is the same 40 nanometers. This is sort of like a pretty good semiconductor chip. This is not like the state of the art, the best that we can do, but when you zoom in into the computer chips and the kind of nanotechnology that we do in computers, these are sort of the fins in between the I don't know that much about semiconductors, but this is sort of when you get it, when you get down to it, like between that there's nothing, and inside that there's a 30,000 RPM motor in poop. So at Ginkgo, we say, or one of our founders has been quoted as saying, biology is nanotechnology that works. And so at nanoscale, you have this incredible thing that biology is. And then when you zoom out, you also have biology that has spread and scaled in this incredible rich ecosystem on Earth. You have the nanoscale, and then you have continent scale biology when you look at things like the rainforests. And so as we look across all of these scales and all the potential and all the things that biology can do, me as a genetic engineer, Ginkgo as a company that's doing genetic engineering, we kind of look to this sort of misquoting of Arthur C. Clarke saying any sufficiently advanced technology is indistinguishable from nature. So what would it mean for our technology to look more like nature? For us, our technology to be more sort of in context or in conversation with nature, not something that tries to scale and grow and take over everything that we can, like the E. coli in the size of Jupiter, but something that really was in that milieu and really had its place. And so I'm going to show you in the rest of the time, I'm going to show you a few examples of E. coli doing interesting things, being engineered and designed to do different things. I'm going to talk about a couple projects that I've worked on that connect some of these ideas and concepts about nature, technology, and society, and using art in particular as a way to kind of play with some of these tensions that we have between all of these different things. And then that's it. So when we look at biology, what it could do and what we want it to do, I am really inspired by this potential for biology to grow, to heal itself, to do things on its own, but to be in conversation with nature and with other things. So when you ask, like, what if your technology could do different things? What if it could be more alive? What if you had a petri dish of bacteria that was programmed to act as a film? So this is bacteria. This is a petri dish, so it's like this big, with a spread with bacteria that was designed by undergraduates competing in IGEM, the International Genetically Engineered Machines competition that Pete mentioned, to be film. So I think I don't remember which way it is. Like, I think they have some sort of stencil, so they shine red light. The parts that have the red light are clear, and the parts that are dark are black. I think they can actually do it the other way around too, so one or the other. And so this is now, like, these are E. coli that are designed to act differently, to do something for us, to be film, basically. And this is the program that they're running that lets them do that. The reason why we can design biology is because biology runs on this kind of code. This is DNA code. So this is the sequence that encodes the machinery inside the cells that responds to light and then activates that color pigment. And then just putting this inside of the E. coli, that E. coli is growing and doing its own E. coli stuff, but it's also reading this code, booting that up, and sort of running that program and able to do it. And so that metaphor, I think, is a little bit simplistic, a little bit too easy, but it's also one that I think is really motivating. It's certainly brought a lot of engineers to the field. That's what synthetic biology is. That's what iGEM is. It's a lot of engineers kind of thinking about, can I program bacteria the way I program computers? What would that mean? What would that mean for what we can do? And what would that mean for the way that we think about technology? So other iGEM projects, notable iGEM projects, bacteria that can play Sudoku. Bacteria that act as a probiotic sensor in your gut, called E. chromai. This is also kind of an art project. So this is sort of imagined in the year 2030, we'll have a yogurt drink that has this engineered E. coli. They're sensing what's going on in the gut, and they're producing pigment in response to the different kinds of things that they're sensing and experiencing. And then you can have a colored output that helps you understand what's going on inside of your gut. This is work by Daisy Ginsburg and James King, who are designers and artists who were working with an iGEM team in the UK. What if you could have bacteria that could heal cracks in concrete? Maybe they were living inside the concrete, and when they were exposed to air, they could create new kinds of materials to fill in those cracks. You have bacteria that can actually do that same thing, sort of bind sand together, make cement. So this is by a company called Biomason. They're taking sand, mixing bacteria in it, and being able to make cement with a lot less carbon input than typically is used for cement. E. coli do things for us all the time, beyond their sort of natural capabilities and what they do for us in our guts. E. coli make riboflavin. So most of the packages of food, they have like nutrition facts, and they say like how much riboflavin, vitamin B2. Almost all of it is made by bacteria, E. coli, that are genetically engineered. A lot of insulin is made by, I think most, or I don't know if it's still made in E. coli. Anyway, insulin can be made with E. coli. So a lot of medicines are manufactured using genetically engineered bacteria and human cells and all kinds of cells. Another iGEM project, iGEM project from 2016, they engineered E. coli to not smell like poop, instead to smell like wintergreen or bananas. They called it the eau de coli. The instructors for this team in 2006 went on to found Ginkgo in 2008. And at Ginkgo, we've actually, interestingly, some of the earliest work that they were doing sort of in a commercial way with some customers of Ginkgo's was to engineer yeast primarily to produce scents for the flavor and fragrance market. So we're working with flavor and fragrance industry to manufacture ingredients, things that smell like peaches and coconuts and oranges and all sorts of different smells, using yeasts that have a program in them that's that genetically engineered program, that DNA that lets them smell like these things. I'm going to give a few more examples. So I have a few more examples of the kind of work that we're doing at Ginkgo, just to give you a sense of the breadth of what might be possible when you can do this kind of engineering. If you can design bacteria, what could you do? Could you imagine bacteria that live in the soil and live on the roots of plants and provide nitrogen fertilizer to them so you don't have to add chemical fertilizers? There's already bacteria that do this for plants like soybeans and other legumes, but we can't do that with other cereal crops like wheat or rice or corn. So Ginkgo and Bayer have a joint venture, another company called Join Bio, working to engineer the bacteria that live in soil and can work with wheat and corn and those other plants and provide nitrogen for them and decrease the amount of nitrogen fertilizer that we use in agriculture, making it more sustainable. We've also got a really interesting project. We're working with a company up in Canada where marijuana is legal, or cannabis is legal, where we're working with them to manufacture the active ingredients, the active molecules from cannabis in yeast. So you can get a high purity and high quality and quantity of many of the rarer cannabinoids that you have in cannabis. So not just THC and CBD that you may have heard of, but a lot of the other molecules that have really interesting properties, whether they're fun or for more of a health or wellness applications. This one's really interesting too. So Synlogic is a company based here in Cambridge. We're working with them, collaborating and helping them. What they do is engineer the E. coli that live in your gut and to act as living medicines. So these bacteria can replace and missing enzymes in your own metabolism. So if you have sort of a genetic disease where you're missing an enzyme that makes it hard to digest something, or then you can replace that with these bacteria in the gut. So overall, the kinds of things that I'm interested in, the kinds of things that we do at Ginkgo, the kinds of things that synthetic biologists look at are really about finding ways where microbes in particular can transform the way we do things in industry, whether that's medicine, like Synlogic, how we approach agriculture and the inputs that we do, like a project with Join, or the way we manufacture things, whether they're medicines, vitamins, flavors and fragrances, or anything else. My job at Ginkgo, I don't work in the lab anymore. My job now is really thinking about those contexts outside of the lab. Where do those microbes live, and how do we sort of use them? How do we make sense of them? How can we design things that kind of collapse these splits that we've made between nature, society, and technology? I think when, we often think, I started kind of talking about how nature and technology are defined as opposites of each other. Often when we, in the science world, we're often thinking about science and society. They're sort of separate, where there's a separation between what we do in the lab and how it sort of is translated to society. In my work, I'm always thinking about ways that to collapse that difference and sort of show that there never was a separation. We're always working together. We're always influenced by society, and that the things that we're doing are kind of a mess and a tanglement in between. So I think a lot about how to communicate about science. I think a lot about how to communicate both ways, like how to engage with people and understand what they're thinking, how they perceive the technology and its potential, and to imagine new kinds of futures that help us better understand what the technology can do. So I want to give one example of a project, the one that Pete tried to spoil for you, because I think it's a good one to show like how microbes themselves are cultural actors, and how this kind of like work in the world of art and culture can help us better understand our sort of future with biology and biotech. So I want to start with a tale of two E. coli's. The first one, this happens like often. So like, there's been a recent example. I think when I first made these slides, it was like Sprouts or something had an E. coli. Like recently it was lettuce. I couldn't find a lettuce. Keynote didn't have a lettuce clip art. But so there's often these moments where when you hear about E. coli, almost always it's in the context of some kind of like poisoning or outbreak or some kind of an event where the bacteria are harming people. There's bacteria are present where they shouldn't be. They're on the vegetables. They're hurting people. There's kind of outbreak. And so that's one kind of E. coli, and then one kind of way that we understand E. coli. E. coli is the kind of toxic invader. On the other side, so this is actually not the best image to reflect the story I want to tell. So there's another kind of E. coli called E. coli Nissl 1917. It's actually the kind of E. coli that Synlogic uses when they do their experiments and the drugs that they're making out of E. coli. It was discovered in 1917 by Alfred Nissl, who was studying a soldier who in World War I was the only soldier in his troop to not get dysentery. And so he's like, something's going on with this guy. Like, he's protected from this dysentery. So he took the poop of the soldier and sort of separated out all the different species of bacteria he could find. And he kind of did a bacterial thunderdome experiment. Like, he put like the dysentery bacteria and the one from the poop, and it's like, 2 bacteria enter, 1 bacteria leaves. And almost always the dysentery bacteria would win, except for this one strain of E. coli that he found. And that E. coli was actually able to kind of dominate the culture and take over, do that kind of exponential doubling, and block the growth of the dysentery. And so Nissl hypothesized that bacteria in the gut of that soldier was actually like protecting his gut and that niche from that bad bacteria from kind of taking over. So I think that this is a really interesting story because I think it shows the two ways, like, when we think of a bacteria as good or bad, and we ask, like, what makes a bacteria as a good bacteria or a bad bacteria, there's 2 things that really matter. One of them is genes. So what's going on inside of the DNA of those different E. coli? The E. coli that's causing that kind of outbreak in the vegetables, that has genes in it from dysentery-like bacteria that's been sort of transferred horizontally from one bacteria to the other, that makes that E. coli more virulent than other more harmless strains. The other thing, though, that's important is context. What else is going on in the environment of that E. coli that's protecting or that's stopping one or the other from growing? How are the bacteria interacting with each other, where that Nissl bacteria from the soldier might stop the dysentery bacteria? So if you had that in your gut and you ate that tainted lettuce, you'd be fine because you have this big sort of diverse ecosystem and a stronger ecosystem. That context is stronger and affects what happens in the population. So that's stuff that in my research as a student and in postdoc, I was really interested in this question. What makes a bacteria good or bad? Could we look at the DNA? How can we shape and strengthen the context of different kinds of ecosystems in order to have a better better kind of situation, better environment. And that had been on my mind when I joined up with a research group called Synthetic Aesthetics. So Synthetic Aesthetics was a program run out of Stanford and Edinburgh, pairing artists with scientists and synthetic biologists to kind of explore some of the ethical and aesthetic dimensions of synthetic biology, and ask questions about like what are the implications of this? What's going to happen in the future, and how can this kind of engagement between the arts and sciences help us sort of think better about the impacts that it might have? And so I was paired with a smell researcher and artist named Sissel Tolas, and she's really fascinating. Her work is really about kind of capturing sense and challenging people through smells to think differently about their environment and how we interact and communicate with our environment. And while I was thinking about what makes bacteria good or bad, a question that really sort of shapes a lot of Sissel's work is the question of what makes some smells good and some smells bad. And so in Sissel's mind, she says, nothing stinks, but thinking makes it so. For her, all smells are neutral, but we ascribe that kind of emotional valence. We say whether or not they're good or bad. And when you look into the sort of literature of even like how we make those decisions about whether it's good or bad, there's certainly something like the genes that the chemistry of the molecules affects how we smell it. This is isovaleric acid. It smells like stinky feet. And so like that sort of experience of the stinky feet like is based on that structure and how we experience it. But there's also a context too. So the way that we experience smells is shaped by the context and the emotional experience that we're having when we smell it. So if you take the same molecule, if you put isovaleric acid in two bottles, maybe you like make them look a little bit different to trick people, you label them differently. You say one is stinky feet and one is Swiss cheese. People are going to have very, very, very different experiences of those same smells. Even if it's chemically identical, you're going to feel different smelling it. You smell the feet and you're like, that's gross, I don't want to smell that. And then depending on whether or not you like cheese, you're going to have a fairly positive, probably, experience of smelling the cheese. And so this is actually, there's a paper about this where they call it an olfactory hallucination. Like your experience of the smell is fundamentally different because of that emotional experience and what you associate with the smell. And so what I find to be there's a few things interesting about this, right? Like our experience and our emotional context matters a lot, and our cultural kind of things that we ascribe to them, like what is clean and dirty and good or bad. But also, scientifically, like why do feet and cheese smell the same? Why is the same molecule there? So as we were sort of like researching and studying and like looking into like stinkiness and bacteria and studying, we just kind of kept finding these overlaps between cheese and feet because those molecules were there, because they have the same bacteria. The same bacteria that live between your toes live on cheese, break down the kind of oils that are either in sort of milk fat or in your skin, and they make the same kinds of molecules. So in fact, there's a professor at Tufts who's a close friend of mine who studies cheese microbiology, and he told me, he says, cheese is about 20x concentrated feet when you look at sort of the chemical and microbial composition. So we are what we eat, but we also eat what we are. And so this was really fascinating to me, and to me at the time, as a student studying genetic engineering and microbiology, I was really sort of struck by how my own feelings of sort of like love for bacteria were challenged by thinking about the bacteria on feet, and how this kind of emotional experience was something that I was also going through as I was sort of imagining cheese and feet, and that discomfort was something that Sissel really encouraged me to lean into. She has, she makes really stinky things, so I'll just do it like as an aside. Like if you go to Sissel's house in Berlin, her lab is right there, and she'll sort of like bring a bunch of smells out, and she's like, smell this, it's cotton candy. Like smell this, it's dog shit. And like smell this, it's David Beckham's feet. And you're like, it's like totally. But anyway, but she wants you to kind of smell that stinky and really to challenge yourself. And so what we did as an artwork to really kind of to I don't know, find our way through this and kind of help people understand this and think about bacteria in new ways, we made our own cheese. We took some samples from people's bodies. We started with our bodies and the people in the lab. So we just got some sterile Q-tips that I had in my bench. We went across the street and we got some milk. We put the Q-tips in the milk and we made some cheeses. We got better at making the cheese over time. This one is actually from a few years after the original cheeses that we did in my lab. This came from the bacteria sort of swabbed from this part of your nose from the curator of the Serpentine Gallery in London, who's friends with Sissel, who sent us in some samples. And so we made all of these different cheeses, not to sell, so like to get a lot of emails asking me like for whatever. I'm not going to make cheese. I don't actually want you to eat this. I did try it though. It's fine. It's food for thought, and it's for thinking about. It's about thinking about bacteria, about disgust, about the boundaries that we draw between what's good and bad, about sort of thinking about how we accept bacteria into our lives in different contexts, in different ways. I think 10 years ago when we started working on this project, stinky cheese was cool in some places of the world. Sticky feet, still not very cool. But maybe the skin microbiome is kind of cool now. People are kind of willing to accept that we're not just human anymore, right? We have these bacteria around. We're rethinking our own health in the context of these sorts of ecosystems. As a genetic engineer too, and one who's sort of part of the synthetic biology community that was thinking all about like computers and programming and gears and cells like that, the work of making cheese also shaped and changed the way that I thought about how we might care for and work with new biological technologies. Because cheese making is a very physical practice. It's very, so this is why you might think that you might realize that human bacteria get into cheese all the time. This is a photo by Rachel Dutton, who's a professor at UCSD who also studies cheese microbiology, and she was a sort of early advisor on the project. But I think what's so interesting when you talk to cheesemakers, they have this real love for cheese and for the bacteria that make it happen. They're really about they're nurturing and kind of farming the cheese. Those bacteria, they're creating an environment where the good bacteria can really flourish, and you can create those kinds of flavors. So to me, I saw cheese making as a really a new way of thinking about biotechnology. This is biotechnology, right? It's using biology to transform something and to make it more valuable and more interesting. And it's really kind of rooted in a love and a care and a nurturing and in a sort of creation of that kind of context. And so those are things that I've really kept with me throughout my career and since doing this project and sort of into my work at Ginkgo, where I think about what are the ways that we can kind of bring that sense of care and love to the bacteria and to the things that we work with, and to our sense of nature. So I don't actually have a good transition here, so I'm just going to go for it. So this is another project. So this is a project I've been working on with Daisy Ginsburg, who made the little colorful turds, and Sissel Tolas again. So we've been collaborating on this project we call it Resurrecting the Sublime, about bringing back the smell of flowers that have gone extinct. And so this is an animation that Daisy's studio did of. This is the slope of a mountain in East Maui, where in sort of the early 1900s, European colonists came and they introduced a lot of cattle and other kinds of animals and for grazing, and they turned all these beautiful landscapes into cattle farms. And that introduction of this other biology and the transformation of that landscape by the colonial kind of overlay into Hawaii really destroyed these forests, and this flower, it's the Hibiscadelphus wilderianus, it went extinct in about 1912. And so that story, or like the loss of biodiversity, the loss of these plants due to human activity, due to human sort of imposition of other kinds of biology and other ways of sort of engaging with biology into other landscapes, is something that I find very sort of difficult to sort of figure out yet. But we wanted to sort of explore what happens like when these things are lost. How might we re-engage with them? How might we bring them back? And in bringing them back, like rethink and reconsider our relationship to nature. And so that hibiscus was. This is a sample, a photograph taken at the herbarium just down the street. So this is a sample from the hibiscus plant that was sampled in about 1910, I think. We were able to find this in the stacks at the herbarium and sample a very, very tiny fragment of the leaves. And at Ginkgo, we could sequence the DNA from those leaves. In collaboration with the UC Santa Cruz Paleogenomics lab that helped us isolate the DNA and separate it out of the tissue. This is what the sequence data looks like when it comes back. It's all these little tiny fragments that we had to sort of, I worked with a bioinformatics colleague named Ju Wang, who was able to sort of reconstruct those and create these new gene sequences that are sort of hybrids of all of these different tiny fragments to put together the smell, the gene, the program that would encode what that flower might have smelled like. And in synthetic biology, this is literally what it kind of looks. I know it's not literally an email; it's like an API. But you send that sequence of DNA, to a company called Twist Biosciences, and they can synthesize it. They can use chemistry to print that DNA from that computer sequence, and we get it in the mail a few weeks later. We have a bunch of robots and a huge lab where we can take that DNA and transform it into yeast cells. Those yeast now have that little piece of this lost, extinct flower growing inside of them. And we can grow those yeast in a tiny, sort of tiny brewery and can capture some of the scents and the smells that they're producing. And we use instruments like this mass spectrometer to identify what those molecules were and what they were making. So we have inside of this yeast this sort of small snippet of that lost plant to bring back and recreate an enzyme that's kind of like this hybrid, not quite alive and not quite dead sort of piece of biology that we can use to now understand a little bit of and recapture a little bit of what it might have smelled like. And this is sort of what the data looks like when it comes back. And so what we did is we got sort of a list of I think it's about 11 molecules from the hibiscus, and I sent that list to Sissel, who used a lot of her skills in sort of identifying and capturing and collecting and composing smells to create a handful of different smells, sort of sketches that she sort of made to reflect and represent what that flower might have smelled like. So I have 3 of those sketches with me. So maybe afterwards, if people want to smell, you can come up and smell. And we're working with Daisy to kind of bring all these things together, all these elements, into an installation that's kind of, the idea is to make it sort of reminiscent of the Natural History Museum and the experience of the human kind of bringing together nature and technology and history and the past and the present and the future. And so it's opening at the Pompidou next week. There's a big show about biology and technology in France with a lot of different pieces, which is really interesting, actually, that there is. Anyway, there's a lot of really interesting art right now that's happening, bringing together these ideas and concepts from art and technology. But you can go smell it in Paris. This project, it's been something that I've been working on this project for many years now, sort of at Ginkgo early on, from this question of is it possible? Can we do it? Can we revive a smell of something that went extinct? What would that mean? What would that look like? How might that sort of help us think about the technology and what's possible as not just, the thing that makes ingredients, but something that can actually capture our attention, but also something that also helps us think about the potential of our own impact on nature and how it might be different in the future. And so again, I have like not a really great transition, but I think I'm also I'm also really interested in kind of this question now of genetic engineering and GMOs, right? Like I make GMOs for a living, and I want. I do love them, and I want people to love them. I think when you think of anyone who's even like remotely skeptical about GMOs and sort of like all the drama and bad things and complexities of what they've done and how they've been talked about might see this and sort of think that I'm being glib or sort of like not caring about all those concerns. And it's not true. When I say I love GMO, I'm saying it because like I care about what they're doing. I want them to be good. It's tough love, right? I want to make good GMOs, and I want them to shape up, right? And I want GMOs that are going to, that are going to do good things. And so I also have stickers. I have about 100 of these, so everyone can have one if you want. So you can talk about what you love and don't love about GMOs, and spark your own conversations with these. This is something I'm like trying to work out and thinking about. There's a philosopher of science named Bruno Latour, and he talks about, he's written an essay called Love Your Monsters, and he talks about Frankenstein. And so Frankenstein is this, is like that quintessential, like, the story of hubris and technology and, the mad scientist who's just wild with power, and he's like bringing back this horrible thing, right? You talk about GMOs, right? It's frankenfoods, right? It's these horrible things that we don't, all we think about is whether we could and not if we should. Anyway, so Latour in his essay he points out, right, like Frankenstein is this monster that we've created and that represents that hubris of technology, and the sort of glib again is like, well, Frankenstein's not the monster, Frankenstein's the doctor, right? He's the scientist, right? It's the scientist who's the monster, and so what Latour points out and what I really want to get at, which is about this love, is he says what he did was, in his sin as the monster is not in creating the monster, not in creating this creature, but in abandoning him, in turning his back away from him and letting the monster go off and roam and terrorize the countryside, because he had been abandoned and sort of felt neglected and lashed out. And so Latour encourages us to think about the future of our relationship to nature as one that we have to be always kind of active and entangled with nature and technology and politics, but we have to love it. We have to be there. We have to nurture it along. We have to follow it. We have to kind of be there to when it falls down, so that we can be engaged and make sure that it's doing good. So that's what loving GMOs means to me. And to me, the other piece of it too, like the other piece of the love is also like the values and the values and the care that you put into the design. So I think often, especially today, when we think about like all the problems in Silicon Valley and how we kind of think about technology, we have this sense of like, well, engineering is neutral or technology is neutral. It's how it's used, or like how it's used when it's out there, and that's not something that we control, right? Like, bad people can use Facebook to do bad things, but that's not, that shouldn't be, Facebook that's blamed for that. And I don't think that's right? Because that again, sort of like, that's Facebook turning its back on that technology. It's not being there and loving it and nurturing it and figuring it out as it goes along. And so rather than a frame of sort of engineering and one of this kind of like arm's length and letting things go off into the world, I call it design. This title is called, the title of the talk is Designing Living Things, because when you design, you're always saying, your values are always there. There's good design and then there's bad design, right? So you're always asking that question. It's a subjective thing. Do you think it's good? What do I think? Is it good or bad? And so I think that's also an important frame of mind for me as someone who's sort of becoming more of a designer rather than an engineer in a company that's looking to engineer biology and thinking about the kinds of products that are going to be out in the world and where they're going to go off after us. I have one last thing, because I think I would also be like, we should when we're talking about designing living things, like we also have to talk, I think, about how we talk about designing people, because it's something that it's always there. There's this like kind of like specter of eugenics and designing people, designer babies, whenever we're talking about genetic engineering and CRISPR. And I think it's easy for us in the field to always be like, oh, well, that's not us. We're not going to do that. That's bad. Don't do that. But I think it is something that we need to engage with and kind of think about how do the narratives and the stories that we tell about genetics and genes and DNA and its power, how do they influence our thinking about what how people should be, how we might engineer them, and all these kinds of things. So I've put these up here because I think you might notice something that's similar between all of the babies. I think when we talk about engineering people and designer babies, it's a very, very narrow set of traits that people are talking about, right? So, there's the beautiful blonde-eyed, blonde, blue-eyed baby, right? It's a very, very narrow sense of what is beautiful, what is desirable. You talk about like, oh, the genes that let you be strong without lifting weights, or like be tall and and it always begs the question, like, better for who? Like, who is benefiting? Is it like who is normal? What does it mean for all the rest of us, right? To think about that. So I just want to kind of point that out as saying, like, I think more than the kind of ethical like catastrophe of actually doing experiments on humans, which is appalling, is the kind of stories that we get as a society about what is normal and what is desirable and who should be, what we should be wanting in ourselves. And so for me, the work is always in kind of bringing in more voices and bringing in more ways of thinking. So we can't be having a conversation about the future of humanity that includes only people who look like this, right? And so this is the last thing I'm going to leave you with, because I really love this quote, and it helps me sort of understand the work of diversity and inclusion in science and technology, and also the work to like bring in some of these concepts from that I've been talking about. So this is Ursula Franklin. She was a physicist and a metallurgist and a sort of scholar and historian and commentator, philosopher of technology. She wrote a really excellent book called The Real World of Technology, and really engaged with a lot of these issues. Very highly recommended. But she writes about sort of specifically the kind of feminist take on technology, and she says, the great contribution of women to technology lies precisely in their potential to change the technostructures by understanding, critiquing, and changing the very parameters that have kept women away from technology. Only then do we have the real possibility of changing the real world of technology. So to me, like, certainly we can expand this beyond just women and talk about, like, people of all different races and abilities and ages and all the kinds of people who are excluded from the narrative and the discussions about and the work of technology, because we think of technology as something that is separate from nature. We think of something that is separate, that's not about care and nurturing and love and emotion and subjectivity. Those issues, those things that are associated with women, they're associated with design. They're separate. So the more we can bring in those other kinds of voices and ideas, the more we separate. Like stop thinking about these things as separate, I think the better technology can be. And so, so that's all I want to leave you with today. To just think about like how things grow and how we might nurture them to grow in a better way. So, thank you. Happy to take questions.